Here we are back in the lab. What we're gonna do back in the lab is we're gonna analyze your food. By analyzing your food, we're gonna find out which macromolecules are in your food. Is it a protein? Is it a lipid? Is it a carbohydrate? Which is, if you'll remember, monosaccharides and polysaccharides. What have we got going on here? Now, you don't really measure nucleic acid because that's in all of your food, because anything that was ever alive has DNA in every cell and yada yada, we go with that. But that's animal meats and stuff like that. Or plants. Plants have DNA in them too. All right, so what we're gonna do today though, is we are gonna analyze some food samples to see which macromolecules they have. Do they have simple carbohydrates or monosaccharides? Do they have complex carbohydrates or polysaccharides? Do they have protein in them? Do they have fats in them? Do they have, what do we see when we come at it? Now, towards that end, I have prepared some food to be tested. All right, please note, here I have some bread. Here I have some pepper jack cheese, because it's pepper jack cheese, and if you're gonna have cheese, why would you not want to have pepper jack cheese? Salami. Salami's good. And then I have, believe it or not, that is chocolate chip. So I crunched up the chocolate chip into little tiny pieces so that it could be dissolved more easily. Now what we are going to do is we are going to take these things, we're going to put them in test tubes with some water to help dissolve and to absorb things. And then once we get it mixed up together with the water, we're going to go ahead and add some of these indicator chemicals. What these indicator chemicals do is they show us whether or not a simple carbohydrate or a complex carbohydrate or a protein is present in the sample. Now we're going to take that, we're going to put a little bit of that indicator compound with the water, with the food, into the test tube. We're going to test, and we're going to see what has what in it. By the way, here's the proof that it was really used to be regular, normal food before I chopped it all up. The food is in the lab because it's being tested. It will not be eaten. That's not how it works. Alrighty, I will get back to you once I have the food in the test tubes and mixed up. Here we have the test tubes with the bread, the chocolate, the cheese, and the salami in it. I put a little bit of water in there so that you'll be able to so that it'll be able to interact better. What we have going on is I'm going to take each set of 4 and I am going to test them with the indicator chemicals. Now the idea is that in the presence of the indicator chemical, if there are simple sugars, simple carbohydrates, monosaccharides in that particular sample, it will change the color of the indicator chemical. If indeed you do that, you do with one of the different indicators, it does that in the presence of complex carbohydrates or polysaccharides. If you do it with a different chemical, it changes color in the presence of protein. And the idea is that we're going to be testing each sample, each of the 4 types of food, for the different sorts of macromolecules. Over here, I also have some water warming up because the Benedict solution requires heat in order for the reaction to happen. Because that's what's going on. There's a chemical reaction between the macromolecule and the indicator chemical that causes the indicator chemical to change color. So we're going to see what is in which of these types of food. The first test I'm going to run is with the Barrett reagent solution. Please note, corrosive, causes severe burns, may be fatal if swallowed, vapor harmful, danger, poison. So, gloves, the careful use of an eyedropper. Wow, gotta get that in the thing. All right, careful use of a dropper in applying it there. I'm gonna put a certain number of drops in each one of the 4 test tubes to test whether or not they have protein in them. Now, in so doing, I'm going to have to put down my phone, so I will apply the drops, and then I will show you, I will record for you what kind of results I got. Okay. Over here, we have iodine potassium. So, IKI, iodine potassium, and this tests for complex carbohydrates, so complex carbohydrates. A negative test is this reddish yellow color, this lighter orange reddish yellow. A positive test for complex carbohydrates or polysaccharides results in a deep blue color. So when it comes to our friend, the the chocolate chips, we have more of that orange red. We do not see the complex carbohydrates. Over here with our cheese, we do not see the complex carbohydrates. Over here with our salami, we do not see the complex carbohydrates. What we do see is here with the, what a surprise, bread. In the bread there is starches, complex carbohydrates, so you get this deep blue, this deep violet color here, showing that indeed this black almost color of the, showing that complex carbohydrates are there, this deep black color versus the others that are not that same color, indicating a negative presence of complex carbohydrates or polysaccharides. All right, I mentioned that when we test using Benedict's solution, we need to warm it up. So what we have here is we have our 4 test tubes with the Benedicts in it, searching for simple sugars or monosaccharides or simple carbohydrates. Basically what I'm doing now is I'm letting them warm up for a period of time to see if the chemical reaction occurs in them to indicate that, that there is indeed a simple sugar in them. Okay, here's our Benedicts. It's been a significant elapsed period of time. Please note, still we're looking at sky blue. Now, because these are all sky blue, that would indicate that indeed they do not have any simple sugars in them. But one possibility is that since the Benedict is already blue colored, the negative might be a false negative. What if the indicator chemical is no longer doing its job? What if it's no longer effective? So I'm going to test that. I'm going to take the indicator chemical and I'm going to put it in with something that I know is a simple sugar. Here we go. So what I have here is some glucose solution, and what I'm going to do is, see, it's not flammable, it's not a danger to health, and it's not reactive because it's sugar water. Anyway, so this glucose solution, it is a simple sugar, so it should react with the Benedicts. So we'll find out whether or not my Benedicts is out of date, and that's the problem, or whether or not it still detects what it's supposed to detect. So this will take a little bit while I have to heat that up as well. I have put the glucose here in this center front part right here. That one's the glucose. And so it is, it's in there, it's getting warm. We'll see what it does and whether or not the indicator works. All right, guys, so look. It's orange, it's simple sugar in the glucose. The glucose is simple sugar. We have liftoff. So I used a higher amount of temperature on that. So what I'm doing is putting these back in at a higher temperature. I'll give them about 5 minutes because if it's much more than that, it might actually break down the complex carbohydrates into simple carbohydrates, which that doesn't help. But we'll see whether or not any of these guys score with a simple carbohydrate with more heat. So cool. That indicator worked, and here's where we get to the barrettes. And the barrettes, as you'll notice, did nothing except slightly dilute. That the chocolate chip is slightly brownish, that the bread is a little bit yellow from the dye in the bread, that the cheese is a little bit white from the dye in the cheese, and that the salami is a little bit murky from the fat floating in it, but it hasn't changed color. So that did not work. The problem here is we really don't know whether the issue is that the barrettes is not actually working anymore, that the indicator solution has gone bad, which is certainly a possibility. So really, or whether or not I've somehow done something wrong in the way that I did the mixing, that I somehow, this is starting to boil, probably shouldn't have that happen. All right, that there was something that I did wrong in the mixing or in the preparation that didn't allow it to properly interact. So the proper way to kind of deal with this is to come back, to order some new barrettes, and to see whether or not using fresh as opposed to multiple year old indicator is really the issue that will fix it. But for what we have found, There are definitely complex carbohydrates in the bread, but not complex carbohydrates in the cheese, in the chocolate chips, or in the salami. That over here, that the Benedicts definitely showed that there were simple carbohydrates in the simple glucose solution, and what we're finding now over here, as we're getting the heat moved up, is that in the bread, guess what? There were some simple sugars. Not necessarily all of it, but definitely some simple sugars. And we're going to find out whether or not there are simple sugars in the cheese, in the salami, or in the chocolate chips. In summary of our little experiment, what we are doing is we are trying to detect different macromolecules in our food, to detect complex carbohydrates or polysaccharides, to detect simple carbohydrates, monosaccharides, to detect protein. And basically, in looking at that, we had 3 different tests. The one for complex carbohydrates went extremely well. The bread registered, wow, there's complex carbohydrates everywhere, but not so much with the cheese, or with the chocolate chips, or with the salami. The protein test failed, pretty much probably because there's something wrong with our indicator chemical, but further research is necessary in order to figure that out. That the Benedict's test for simple sugars, it did not work originally on our samples, our 4 different samples of bread and chocolate chips and salami and cheese, but when tested with a glucose solution, glucose being a monosaccharide, a simple sugar, it went ahead and turned the proper color for the indicator. At which point we boiled at a higher temperature the bread. What happened is the bread, then the complex carbohydrates in the bread broke down and began having simple glucose be a part of it. Remember, one of the things that your body does with the complex carbohydrates is it breaks the bonds between the individual sugar molecules, so that, for instance, when you in cheese have the lactose, it breaks down into glucose and galactose. It has to basically chop it apart so that those monosaccharides can pass through the intestinal wall. Same thing here. Basically what happened is the boiling water was digesting the bread and separating it from these complex carbohydrates, pouring energy into the chemical bonds so they could be broken, and then getting it into being individual monosaccharides, which then reacted with the Benedicts.